Welcome. Thank you all so much for joining us today. My name is Drew Scammell, and I'm a program associate with the Inclusive STEM Ecosystems for Equity and Diversity, or IUSE program at the American Association for the Advancement of Science. On behalf of our AAAS IUSE initiative, I'd like to welcome you to today's workshop, Forest-Based Undergraduate Research Experiences, or CURES, incorporating STEM research into the curriculum. Today's workshop will feature presentations from Dr. Emma Goodwin and Dr. Oliver Hyman. Before we jump into the workshop today, I have a few housekeeping notes I'd like to go over with you all. Go next slide. This presentation is being recorded, just so you all know. The recording will be made available in the coming weeks on our website at AAAS-IUSE.org. And the slides are posted on the website and available in the comments below if you'd like to follow along. You'll also receive an email with a link to the recording when it's available. Please note, we will transition to a Q&A discussion breakout following the presentations, and these breakout rooms will allow for deeper engagement and will not be recorded. So once both presenters go through their presentations, you'll have the option to attend either of their breakout rooms, and those will be non-recorded discussions. Additionally, we have closed captioning for this workshop, which you can download and view the full transcript by clicking the CC button at the bottom of your screen. And for those of you who are new to the AAAS IUSE initiative, this initiative seeks to support faculty, students, and the greater undergraduate STEM education community by disseminating research and knowledge about STEM teaching, learning, equity, and institutional transformation. We invite you to learn more about the AAAS IUSE initiative on our website and hope you will join our community as future contributors. And at the end of the presentation, we'll go over some upcoming searches that y'all will hear about in the future. You can also follow us on X, or formerly Twitter, at IUSE Program, and LinkedIn at the AAAS IUSE Initiative to stay up to date on our latest events, blogs, or to share what you've learned at today's workshop. And with all of that out of the way, we'll shift over to our first presentation. So without further ado, I'd like to welcome Dr. Emma Goodwin, NSF STEM Education Research Postdoctoral Fellow at Arizona State University. So Dr. Goodwin, I'll pull down my slides and I'll shift it over to you.
Perfect. All right. My slides look good there? All right, awesome. So my name is Emma Goodwin. I'm a postdoctoral researcher at Arizona State University as part of the Research for Inclusive STEM Education, or RISE Center. And I really appreciate the opportunity to talk to you all today about CURES. So today, we'll be going over why we should incorporate CURES into the undergraduate curriculum, what makes a CURE real research, things to consider when teaching a CURE and when designing a CURE, and whether we know if CURES actually provide equitable research experiences for students. And I have a lot of citations throughout this talk. I've provided many of them on a worksheet that you should be able to access. So to start, we know that engagement with undergraduate research can result in numerous benefits for students. These can include support in motivation in science, increased scientific self-efficacy, increased interest in science, sense of belonging in STEM fields, and ultimately even retention in STEM majors and careers. And because of this, there have been multiple calls for all STEM undergraduates to engage in research experiences. And traditionally, research experiences in faculty mentored research labs, a student is a member of the research lab. They're contributing to a lab research project. They're mentored by senior members of the research lab, maybe the PI or grad students or senior undergraduates. And there are multiple problems with this model, right? So there are a limited number of positions available in faculty member research labs. And at most institutions, there's more students enrolled than positions available. So this is a problem if we want to meet that call of getting all students involved in research. And then there's also a selection process in who gets to participate in undergraduate research. So students need to know the unwritten rules of undergraduate research, how to actually get into that experience. And they need the confidence to apply and the time to add research onto their normal coursework. And there's also the issue of selection bias from faculty, because faculty are incentivized to take the best students who are going to be the most productive researchers, who are going to go on to grad school or med school. And faculty implicit biases may advantage certain populations of students. So we know faculty are more likely to respond to emails from white sounding names compared to black sounding names. There's research that faculty perceive men as more competent, hireable than women candidates. So these issues can kind of be boiled down into two overarching problems. So first, only a subset of students are able to participate. And second, these students tend to be from more privileged backgrounds. And one way we can address this is to create initiatives that are diversifying who does research in faculty mentored labs. And this solves that, or at least addresses that second problem, but it does not address that first problem. So through integrating real research experiences into undergraduate courses, we're able to really address both of those issues. Because high enrollment cures, course-based undergraduate research experiences, or many low enrollment cures can increase the number of research opportunities for students. And then there's greater equity in participation, because if students are required to enroll in a cure as part of their normal curriculum. Students don't need to know those unwritten rules of how to enter a research experience, and faculty don't need to select the best students. So there's many different terms that are used to describe undergraduate research as part of a formal course. We're discussing cures today. And what makes a cure a real research experience for students? So we've talked a little bit about faculty mentored research experiences. In cookbook or traditional lab courses, students are following instructions to learn scientific principles or lab techniques. So really engaging in those confirmatory activities. Inquiry courses can include a wide range of instructional experiences, but generally students are conducting experiments to address scientific questions. And in all four of these experiences, students do have the opportunities to engage in scientific practices, use tools and techniques, and they often have the opportunity for collaboration. So they're working with other students or research mentors or their instructors. And in inquiry courses, cures, and faculty mentored research, we start to have more opportunities for students to practice iteration. So this idea of doing a project that's building off of previous scientific knowledge and also having opportunities to repeat experiments, to adjust things and move on from there. In cures and faculty mentored research, we start to see opportunities for novel discovery. So this means that, and this can even happen in advanced inquiry courses as well, but this means that the instructors, the students, no one knows the answer to the research question that they're trying to address. And it's not something they can just Google. And really specific to cures and faculty mentored research, students are addressing a question that has an audience outside of the classroom. So there's a broader relevance or a community impact or some level of importance that extends beyond that classroom environment. And another way I like to think of these experiences is that the purpose of conducting cookbook or traditional lab courses and inquiry courses is to give students opportunities to simulate scientific research, to experience the process of science. But in cures and faculty mentored research, a student is actually participating in research. So they're attempting to generate products and answers to scientific research questions. So ultimately, it's these last two elements, broader relevance and novel discovery, that we tend to say makes an experience a real or authentic research experience for students. And sometimes we say broadly relevant novel discovery or just relevant discovery. But what we mean by that is that students are engaging in research that has the potential to produce novel findings with an impact beyond the course. So we have talked about what we think makes an experience a real research experience. But we're going through a lot of trouble, right, in implementing cures to give students an actual research experience. So it's important that students are actually realizing that they're engaging in real research as well. So I'm going to stop talking for a few seconds. I'd like you to imagine that you are a student in a cure. As a student, what would make you feel like you participated in real research? And I'll give you about maybe 30 seconds or so, and I'd love for you to share your ideas in the chat. These are really awesome ideas. So I'm seeing some recognition getting published. So having your name on a publication, just understanding the bigger picture, the purpose. I'm seeing some like autonomy and getting to contribute to research questions. Thank you. Freedom, creativity, acknowledgement. Awesome. Okay. So I conducted a study a few years back to address this question. So we asked students in a CURE what made their experience feel like a real research experience. These were open-ended questions. We coded themes in students' responses, and we actually found that experiencing failure was the #1 response we got from students in terms of what made their experience feel like the research experience was authentic. And I will give you the caveat that students experienced a ton of failure in this class. So that might have influenced the answers, but it was really fascinating. One student said, "I love that the experiment did not go as planned. I mean, it's not ideal that a bunch of our embryos died, but this is how real science works. I am usually so bored in the assigned cookbook labs. They are carefully designed so that students get the right answer." And students also talked about iteration, scientific practices, relevant discovery, autonomy, collaboration, and even experiencing successful science. But what we really took away from this project is that experiencing failure in a lab course, like a cookbook course, or even sometimes inquiry courses, where there might be a right answer, can be really frustrating for students. But in a CURE, when there's opportunities for relevant discovery, failure can actually make the experience feel like real science. And I will highlight that only 3% of our students actually said that successfully answering the research question made their experience feel real. So hopefully that takes some pressure off in terms of having to have a research project that always works out perfectly all the time. So broadly relevant novel discovery, so essentially real research, is also associated with increases in students' project ownership. So in a study that considered both cognitive ownership and emotional ownership for CURE students compared to a traditional lab course, cognitive ownership and emotional ownership were significantly higher. And this is important because project ownership can support students' resilience to challenges of research and failure in research. It can support students' scientific self-efficacy, motivation, and interest in pursuing science careers. So many benefits from real research. Another benefit is publication. So in some CURE, students can have the opportunity to be co-authors on scientific publications. This is also a great benefit for instructors too, who can get publications out of teaching these courses. And in some research projects, a scientific publication just isn't the most impactful outcome, but the work in the CURE can still reach a broader audience by having students contribute to a database or community reports, which could have the potential to change how people think or behave beyond the course. So it has that relevance beyond the course. So some things to consider when teaching a CURE. We have studies that show that faculty instructors of CUREs discuss that teaching a CURE requires this kind of dual role, where you're both being kind of the traditional instructor, supporting students, but you're also serving as a research mentor to develop students as autonomous researchers. So combining that research mentor and instructional role. And successful CURE instructors should be able to deal with uncertainty in terms of not knowing what's going to happen with research projects. each day should have a strong background in research, both the research topic at hand and also just generally the process of doing research. And especially when first developing or teaching a cure, be willing to invest time and effort into the process. But especially at large institutions, many lab courses are actually taught by teaching assistants directly. So this is something we need to consider as we implement cures into courses that are actually directly taught by teaching assistants. So graduate teaching assistants, or TAs, are often novice teachers and researchers. So are they prepared to support students and serve as research mentors in a cure? So as part of my dissertation, I conducted interviews with TAs, and we did find that most TAs do understand this dual role of both supporting students and serving as research mentors. So one TA said, the first time they don't get phage, I tell them, research is 99% troubleshooting, and I give them my example. I've been working on this part of my research for 6 months, and I ended up getting nothing, but I'm still here teaching and smiling, so you guys should not be sad. So you can see that this TA really understands their role as a research mentor, but is also supporting students kind of emotionally. However, we did find that some TAs really do struggle with the unpredictability of research in a cure. This TA said, some students in the cure don't understand what is going on, and they start to believe that I'm not good at teaching. But when it's a cookbook lab course, everything's prepared. When the students get the results that I expect, I'm ready to elaborate and build on what they have seen in the test tube or the DNA extraction. In the cookbook labs, I'm ready for everything. So this TA really struggled with unpredictability in the cure, and actually that felt like a threat to their authority as an instructor. So this can be a problem for some TAs. We've also found that cure students are motivated to engage in a cure when their TA supports their autonomy as novice researchers, provides an appropriate level of support, so is there to provide guidance, but is not there to just tell students what to do and give them all the answers right away. That kind of appropriate guidance, yeah. And also develops a sense of community in the cure. So students talked about feeling like a member of team, and like they were, a part of a research team rather than just individuals in a class, and that was really powerful. So things to consider when designing a cure. So using backwards design to define research and teaching goals can be a really useful approach to designing a cure. So in any course, we have pedagogical goals, right? So we have the concepts we want students to understand, maybe communication skills, and other skills. But in cures specifically, we also have our research goals. So essentially a research question. What do we want students to be able to address in the research in this cure? And it's helpful to start by identifying those goals, those desired outcomes, and then figuring out what is the acceptable evidence that will lead us to understand if we have achieved those outcomes, right? And we need that both for our pedagogical goals and for our research goals. And only once we have that, once we know what our goal is and what the evidence that we need is, does it make sense to really plan the experiences and the instruction and the research experiments that will actually lead us to produce that evidence to meet those goals. So these are outlined in a paper by my colleagues, Katie Cooper and Sarah Brunell. And something else that we like to encourage instructors to consider when designing their own cure is thinking about how you can leverage your own expertise and your own personal goals to support your students. So for the next few minutes, I'm going to introduce kind of our personal example of cures that I've been involved in with Dr. Sarah Brunell. So our research and expertise is in science education research, specifically on undergraduate research experiences. And Conducting research and publishing supports our professional goals, right? I'm a postdoc. I'm on the job market. It's very useful for me to be publishing right now. So we involve students in a science education research care to study undergraduate research experiences. And through this experience, we're able to support our students first, but also we're leveraging our own research expertise and background. And we're promoting our own professional goals because we can publish on our own research with our students, right? So there are some advantages, there are several advantages of CURES, both generally and in science education research. So advantages of all CURES are that students can develop research skills, there's potentials for student co-authorship, and that faculty can often merge their research and teaching interests and responsibilities. Some advantages that are unique or specific to science education CURES are that they can actually support student metacognition about their own learning and experiences. So we're having students develop research and analyze data related to undergraduate research experiences, so students can actually use that to think about their own experiences. They can easily be conducted in person or remote, which has been a huge advantage in recent years. And they have the potential to increase the generalizability of science education research, right? We can do replication studies at different universities with different student populations, which can be really helpful for building knowledge about undergraduate education. So Katie Cooper and Sarah Brunel have also published an incredibly useful paper outlining recommendations for a science education cure. I put science education in brackets because I strongly believe that these recommendations are excellent for every single cure. So I love this paper, highly recommend it. The first recommendation is to identify an accessible and relevant research question. So a question that undergraduates can understand and really buy into. Second, to take advantage of a large number of researchers. So many hands make light work. You're working with a bunch of students. It's really useful to find a project that can actually leverage the fact that you're working with many students. So can they do little parts of a larger project? Avoid the need for high-level technical data analysis. So you don't want to spend weeks and weeks training students to do one certain technique. Build in accuracy checks. This is super important because we want to be producing real usable research, but you also need to be making sure that your students who might make mistakes or maybe won't be as diligent, that they're actually doing a really good job with their research. So having students check each other's work or having systems where the instructor is spot checking data is really important. Flexibility with timing. So you don't want one experiment that goes wrong to derail your entire term. So having some flexibility in the schedule for you to recover from unexpected things is really important. And finally, to stay organized and develop systems to help students stay organized. So maybe these are structured lab notebooks where students are, you're telling students what information they need to be recording and keeping track of. Or other organizational systems. So we conduct our science education here over the course of an entire academic year. We have students help us plan the project, design often a survey or interview protocol. Students engage in data collection, data analysis, and hopefully at the end of the year have a chance to start thinking about data interpretation and the actual manuscript. But in terms of actually putting together and writing the manuscript, that's often led by instructors over the summer, ideally, and we have the student co-authors review the manuscript before we submit. And I'll highlight that completing the project in a single term is possible, but it often comes at the expense of some student autonomy, right? So if you're doing everything in a single term, you might need the instructors to play a bigger role in helping students arrive at that research question a little quicker. So in the early weeks of the cure, when we're planning a research project, we allows students to really kind of have a lot of say in what research questions we're actually answering, but we do tell them that they must align with our research interests. So they must be related to undergraduate research experiences and tied to diversity, equity, and inclusion. So students are coming up with these ideas, but we are giving them a lot of prompts and feedback and engaging in a lot of discussion to help guide those ideas. And then the project must also have potential for broadly relevant novel discovery, right? It must be real research. It must be a novel question, and it must be feasible given our resources and the structure of the cure. So these last 2 points, students have no way of knowing what has already been done. They don't have the expertise to know what has already been done in the field and what is feasible and not. So these last 2 points require heavy guidance and feedback from the instructor. So I'm actually going to address this last question by sharing an example of a project that we've conducted as part of our science education cure. With our students, we wanted to understand to what extent do studies evaluating student outcomes on undergraduate research experiences report and analyze demographic data. This was a project we conducted over the pandemic year when we were working remotely. We had CURE students identify 147 research papers, so they over Google Scholar, 147 research papers on student outcomes from research experiences, and students coded which demographic categories were included. So I will highlight that these are research papers, education research papers on research experiences that we were having students look for. So on the right side, I have all of the demographic variables that we coded, and I'd like you to make a prediction. What percentage of research papers on the outcomes of undergraduate research experiences consider any demographics at all in their analyses? And we're going to do a waterfall chat. So I'd like you to make a prediction and type in the chat, but wait, don't press enter yet. I'm going to give you about 20 seconds and then we'll all press enter at the same time. So make a percentage prediction. All right, go ahead and press enter. Oh, this is amazing. Everyone followed instructions. Okay. All right. So hopefully most of you will be pleased that the actual answer is higher than most of the predictions. So we found that 62% of research papers on outcomes of undergraduate research experiences do consider a demographic variable in their analyses. So that's great. When we look at which demographic variables are considered, we see that this is almost all gender, not almost all, but mostly gender and race and ethnicity. So these green bars can show you that 40% of papers, just over 40% of papers, are considering gender and race and ethnicity in their analyses. But when we consider all of these other demographic variables, we can see that we almost know nothing about the experiences of, say, students with disabilities in undergraduate research experiences, or LGBTQ students in undergraduate research experiences. So there's a lot about student identity and research experiences that we still do not know. So we know very little about how student characteristics can impact outcomes and research experiences. One other thing that we found in this study is that demographics are actually less frequently considered in papers on course-based undergraduate research experiences compared to papers on faculty-mentored research experiences. And so that means, it's the call to action, if you are thinking about developing cures and research experiences, more research is needed to understand if and how cures are providing equitable research participation for all students. So we encourage you to consider how student demographics impact student outcomes in your own cures. And with that, thank you very much. And I think that's our time.
Amazing. Thank you so much, Dr. Goodwin. I'd like to now welcome our next speaker, Dr. Oliver Hyman, Senior Lecturer at James Madison University. So Dr. Hyman, feel free to pull up your slides and take it away. I think you might still be muted.
I was going to see if I could make it through the whole talk on mute, but I couldn't pull that off. Right. So I'm Ollie. I teach at James Madison University in Harrisonburg, Virginia. I'm going to talk to you a little bit about a cure that we're doing here in our large enrollment intro bio labs that uses DNA barcoding as the sort of main model. But for those of you that may not teach intro bio or intro level courses, just rest assured that DNA barcoding is something that you can use at sort of all levels of the curriculum. I'd like to start off just acknowledging that we never could have pulled this off without help from a lot of other folks, especially at the Cold Spring Harbor DNA Learning Center, and also with help from the iPlant Collaborative and the Cyverse, which created this website called DNA Subway that we rely upon really heavily to do DNA barcoding. And of course, support from administrators here at James Madison University to be able to pull this off, and all of our instructors here at JMU, and the National Science Foundation, who helped fund some of this research through the Cold Spring Harbor. So back in 2009, this Vision and Change report came out from AAAS. I'm sure everyone here is probably well aware of this document. It's been over a decade now, and a lot of important reports and ideas came out of this. But one of the big things to come out of it is encapsulated nicely from this quote from Bruce Alberts that basically said, "My hope is we can change the focus of introductory courses to cover much less material in order to give students the chance to learn what science is and the opportunity to experience science." And one of the big models that's emerged to sort of accomplish that goal is this idea of course-based undergraduate research experiences. And we've gotten a really nice introduction to all the advantages that cures offer. And they're sort of loosely defined as novel research that is broadly relevant to people outside of the course. There's a lot of different ways that you can incorporate cures into a course. And one of the biggest challenges, I think, is how do you do this with a large enrollment freshman course where you have many students and many instructors that don't have a lot of experience teaching or doing research. There's a lot of big barriers, both logistical and practical, to incorporating cures into these types of courses. And this is something that we really wanted to try, given all the benefits that we've heard about here at JMU. And the model that we came up with to do this was using DNA barcoding. So a little quick background on DNA barcoding. Basically, it's a technique that you can use to identify species or any sort of piece of biological material to the species or genus level, taxonomically speaking. The way it works is you sample an organism or some piece of organic material that you can extract DNA from. Once the DNA is extracted, you can amplify it through PCR, looking for a specific barcoding region of the genome. Once you amplify that region, you can send it off for sequencing, and that is your DNA barcode. You can take that DNA sequence or the DNA barcode and enter it into a database. You can use either NCBI or the Bold Systems database. There are some other databases as well. And essentially, it's like a Google search for that organism. It'll pop back what the taxonomic ID is, once again, to usually the species or genus level, though not always. This has kind of been likened to Spock's tricorder, where you get a little piece of a sample and immediately spits back what it is. We don't have it this slicked out yet. It takes a little bit more work than that, but it's the same general idea. There's a couple really big advantages of DNA barcoding, especially for an introductory biology level cure. It's really scalable and it's very reliable. It's scalable in the sense that the protocols are relatively cheap, ranging from about $4 to $10 a sample from start to finish, including all the reagents and disposable things that you need. It's predictable in terms of its workflow. So in that sense, it's very useful when you have lots of different instructors with different levels of experience and lots of different sections. You don't have a million different research questions that are happening that all require different equipment and different types of expertise. And also the protocols themselves are simple enough that first-year students and first-time instructors can generate reliable data. You know, at the end of the DNA barcoding pipeline, you either get a good sequence or you don't get a good sequence, and you know if it's a good sequence, it's reliable. It's also really nice in the sense that it's flexible, so you can address a lot of different research questions without having a ton of expertise. And typically these fall into 2 or 3 different categories, usually related to something like product mislabeling. So is this sushi the species of fish that they say it is? Is this pollen actually coming from the flowers that they say it is? It's good for biodiversity inventories. If you have a local taxonomic group or a local area that you're looking at the biodiversity of, it's great for that. And it's also useful for describing underdescribed life stages or underdescribed taxa, or helping to build up barcoding databases from natural history collections. And the other really nice part about it is that it's cross-disciplinary. So you can use tools from a lot of different fields, whether it's evolutionary biology, ecology, taxonomy, cellular molecular biology, and bioinformatics. It's an opportunity to touch on a lot of different areas. And it's also an opportunity to sort of appease folks that may be stakeholders in these intro courses that you're still going to be able to talk about that sort of special thing that must be talked about in these intro courses that can't be dropped from the course content-wise. So just to give you a couple quick examples of some DNA barcoding cures, these can really be either local initiatives or national programs. At the national level, there's an interesting example that the Cold Spring Harbor DNA Learning Center is doing. Basically a citizen DNA barcoding network looking at barcoding of United States ants, where they're basically collecting barcodes all across the United States to build up a big database of the biodiversity of ants across the United States. But it's also flexible in the sense that you can do local initiatives and local projects that are relevant to your particular area and your particular research questions or topics. And so some examples of that might be looking at local sushi restaurants or fish purveyors to see if they're mislabeling their fish products. And also looking at, just as examples, looking at insect diversity and range expansions of things like cicada killer wasps, some invasive species, or potentially looking at the species of mosquitoes that can be disease vectors that can potentially be spreading into new ranges and new areas. And so these are all different examples of DNA barcoding cures that can be relevant outside of the course and contribute to larger databases or individual sort of research programs at your particular location. You can also use DNA barcoding for more of an inquiry style course. So this is less of a cure where it may develop research that's definitely novel to the field, but it is novel to students and may be interesting to the students. And this is basically where students come up with their own research projects. And this is one model that we've tried here at JMU outside of the course that I'll be talking about today. But basically they develop the research question, write a proposal, collect their samples, run through the DNA barcoding pipeline, write up their results, and then try to put together a discussion abstract towards the end of the semester. And these are just a couple examples of the projects that students have come up with in a semester-long research project here at JMU, looking at fungi in dorm showers, pollinators of non-native plants, yeasts and kombucha scobies, pollen in local honeys, the taxonomic identity of octopus at local seafood markets, and looking at shark jerky and what sharks were present in the jerky. My colleague Ray, who's present today and will be here in the breakout room as well, is also leading some initiatives looking at meta barcoding, which instead of normal DNA barcoding, where you get your sample, you extract your DNA, and you put together a barcode to identify one individual sort of target organism, meta barcoding leverages next-gen sequencing to basically look at all of the different species present in a sample. And you can start to look at all of the species and sort of develop questions related to things related like microbiomes, and also looking at maybe fungal diversity in a particular soil sample or region. And just to give an example of the types of projects that Ray's been working on, they're looking at soil and water microbial communities at contaminated versus clean sites, and also looking at male versus female garter snake microbiomes. And here you can see Ray in the background and some of his students swabbing snakes as part of our colleague Rocky's research program. So I want to spend the rest of the time talking about the intro DNA barcoding course that we're doing here at JMU. This is a freshman lab. It's called Bio 140 that enrolls about 500 to 600 students per semester. This is the required course for all of our majors. They all have to take it. In addition, it's a required course for many non-bio, like health science majors. So about 60% of the students enrolled in this course in any given semester are biology majors. The rest are usually some other either health sciences or science major. It's broken into 20 plus lab sections with 24 students per section. Our teaching crew includes tenured faculty, full-time faculty that you can see with the little arrows over their head here, master's students that are teaching for the first time, and then some part-timers that sort of teach out of the goodness of their hearts. And we do have a dedicated lab coordinator for that course that's involved in training many of the first-time instructors and making sure all the necessary materials are present in the classroom each day. So the Bio 140 course is addressing a research question related to edge effects and how those edge effects influence biodiversity at forest edges and forest interiors in an arboretum that we have close to campus. We spend the first 4 weeks talking about some fundamentals, so things like the process of scientific discovery, experimental design, and we talk a little bit about evolution and interpreting phylogenetic trees, because that's important towards the end of the semester when they're doing their bioinformatic analyses in the DNA subway software. The next 2 weeks are sort of ecology module, where students go out and run transects in the forest interior and the forest edge. The students are broken up into groups. There's a fungus group, there's an insect group, and there's a plant group. During this time, they're sort of quantifying all the morphospecies along the transects and comparing biodiversity of species in each of their transects at the forest edge and in the forest interior. And they're also collecting an individual sample that they're going to identify through DNA barcoding. So they'll take their sample back and run through what we call the sort of DNA barcoding workflow or portion of the semester. And so during this time, they'll learn how to pipette. They'll learn about DNA extractions, they'll run a PCR, they'll run a gel, and any successfully amplified products will be sent off for Sanger sequencing to generate a unique DNA barcode. And then the last part is running that barcode through the DNA subway. This is the blue line of the DNA subway, if you're familiar with this software at all, where essentially they take their sequences, they assemble them and clean them up. They now analyze them through a basic blast search, and then they generate phylogenetic trees based on the closest blast hits to help identify the taxonomic ID of their organism. And those sequences can also ultimately be uploaded to the NCBI database through DNA Subway. So they're contributing to a larger database of DNA barcodes. Throughout this, we're practicing scientific communication, so they have lab reports and posters. In addition, there's several assessments in terms of exams and finals, just to kind of make sure that they're learning the material as we go through the semester. The nice part about this is it's explicitly linked to some really important fundamental concepts that are going to be important for all of our instructors as our students move through our curriculum into the upper division. It's also explicitly linked to important techniques that they're going to need as they move through their curriculum. The other nice part is failure and iteration are included in the DNA extraction and PCR amplification portions of this lab. And if anyone's ever done a PCR before, you know oftentimes they just don't go the way you want them to go. So we have extra time for students to troubleshoot PCR, and we teach them a couple great tools and techniques for how to do that. And so students have the opportunity to practice iteration. And the nice part from the instructor perspective is these PCRs, they fail in sort of predictable ways. And so you have a very sort of explicit set of tools that you can operate or you can offer them to know how to troubleshoot that, which is nice for first-time instructors. Also, once again, this does cross disciplines and introduces students to lots of different fields. This is really great for a first-year course because you want students to kind of dabble in all areas of biology to enter, identify what their interests are, but also be able to know, like, this is a thing that I'm not necessarily interested in. So maybe I want to keep on this ecology route, or I really like the bioinformatics part. Maybe I want to explore more courses in the upper division that have applications to that. And the other really nice part is this is all modular. So when we introduced this cure, we basically wiped out all our old labs and put in this giant 14-week behemoth of a course, which is not practical for a lot of people. But the DNA barcoding workflow portion of this, which is really weeks 7 through 13, you can make an abbreviated version of this that you can run in 3 weeks. Or if you just wanted to do the bioinformatics course, you can do that in like an hour or 2-hour lab. So there's little bits and pieces of this that you can break off if you don't want to sort of have to deal with the entire bear of a whole course rewrite. So just to sort of walk you through what this looks like from the student perspective, this is Emma. She was part of the mushroom group. They ran their transects. She collected this mushroom. After she collected the mushroom, she added it to this database, this sort of project that we've put together in iNaturalist. You can see her little sample there with an arrow and a picture of it. So she's tagged it there. She extracted DNA from that sample, and this is an actual Snapchat that Emma and her group members took. They were very excited that their PCR worked, and they got some great bands. You can't tell, they're gel electrophoresis masters. Those products were sent off for sequencing. So basically, they load them up in a tube, and we ship it off to Eurofins. And within a 24 to 48-hour period, they email us back all those DNA sequences for our students to ultimately, in the next week, analyze in DNA Subway. Then they identify their organism. Once they've got a good ID on it, then they plug it back into this database that we're building up here at JMU to look at the sort of total biodiversity at the edge and interior of our campus arboretum. And so here's our barcoding biodiversity at JMU Bio140 database on iNaturalist. You can see that we've got several thousand observations, several hundred different species that have been identified, and you can see some of the most identified or most common species that we found in the area. And if you zoom in and click on any of these, you can see, for example, Emma's mushroom that she found. It's a lion's mane mushroom, and you can see her DNA barcoding sequence for that sample and all the associated metadata that can be downloaded from the website. So we've done some baseline level student assessment. This is a survey from both the bio and non-bio majors here at JMU. And this is just asking them, in comparison at the start of the term, how has the lab experience changed your level of comfort with concepts from ecology, molecular biology, and bioinformatics, and techniques and methods from ecology, molecular biology, and bioinformatics? And perhaps not surprisingly, most students rate themselves as either more comfortable or neutral in terms of their level of comfort with these concepts, techniques, and methods. This is perhaps not super surprising, and perhaps not entirely convincing in the sense that students are not the best at assessing what they don't know. But the real take home from this, and the nice part about this, is this means students are coming away from this not saying like, I didn't understand any of this and this was extremely frustrating and I feel like I don't know anything now. Instead, they're saying, yes, I feel comfortable with this. And so we're pitching it, and this is being pitched at a level that students can deal with and come away from feeling like they've learned something. That is to say, these cures aren't insurmountable at this freshman level of a course. Looking only at the JMU bio majors, this is from a recent semester. This is basically looking more at sort of student affect and how they feel about biology in general after they take this 140 cure. So this just asks, how did the 140 laboratory influence your overall academic or degree interests as a biology or biotechnology major? And I think the real take home from this is from 151 students, and the percent that basically agreed that they were more interested in biology or reinforce their interests, or there was no change in interest, but only 9% are saying they're now less interested in biology. So luckily we're not turning folks away from the major with this cure. A similar question, how did the Bio 140 laboratory influence your career interests as a biology or biotechnology major? And once again, looking at 151 bio majors that took the 140 course in fall 2021, only 8% are saying they're now less interested. So once again, we're not scaring folks away from a career in biology. This is looking at student belonging. So we just prompted students, once again, this is the same fall 21 freshman biology majors. As a result of being in this class, I feel part of or belong to the JMU biology department. And only 8% are disagreeing with that statement. So it appears that this course is also helping to create a sort of sense of belonging within the JMU biology department, which is good. In addition, these are some data that my colleague Joe Harsh and his collaborators have been working on. He was kind enough to share with me. This is looking at science self-efficacy. So this is part of the persistence in the sciences survey that was developed by Dee Hanauer's group. These are 6 validated items related to basically student self-efficacy. So these are the 6 items. Basically, students are responding to, I am confident that I can use technical science skills, generate a research question, figure out what data or observations to collect and how to collect them, create explanations, use scientific literature, reports to guide my research, and develop theories to integrate and coordinate results from multiple studies. And overall, our students reported a mean of 4.15 on a 5-point Likert type scale, from strongly disagree to strongly agree. And so it's hard to say exactly what that means, because unfortunately for a lot of these surveys, we didn't have a comparison group, a non-cure comparison group. In the ideal scenario, we would have ran these surveys with the original labs that we were doing long before we ever changed the courses, but we didn't begin to do any of these assessments until after we had switched our curriculum over to this Bio 140 cure. But because this is a validated survey that other folks are using, we're able to at least compare these responses to those of others. And so this is the CPHAGE program, if you've ever heard of it before. That CPHAGE program has done these same sort of, these same surveys. And looking at a master's institution similar to JMU, their self-efficacy numbers are very similar to ours with a value of 4.1. So we're sort of on par with other well-established cures in terms of self-efficacy. And lastly, we're beginning to look at students on the national level. And so this is a survey of a large sort of consortium of people that are running DNA barcoding cures across the United States that we've helped to train and collaborate with. And so this is looking at a sample of around 1,200 students from 2-year, 4-year, minority serving, and historically Black colleges and universities. They're implementing some form of a DNA barcoding cure. And you can see there's a couple different colors here, ranging from strongly disagree to neutral to agree to strongly agree. And basically, we've asked them some simple questions related to how they feel after completing their barcoding research project. I have become more interested in technology and bioinformatics. I have a better understanding of the importance of technology in modern biology research, more likely to pursue bioinformatics and technology in college than I was before, more interested in studying science in general, more likely to study science in college than I was before, capable of going further in science, and become more interested in studying biology. And the sort of take home from this, I think, is that most of the students are in the agree to strongly agree categories, many neutral, but very few are in the strongly disagree to disagree category. That is to say that this is decent evidence that DNA barcoding and these types of cures can be valuable for accomplishing some of these goals related to sort of student affect and their interest in science and bioinformatics. So if you're interested in learning more, there's wonderful resources at the DNA Learning Center related to their Barcoding 101 webpage, and the website's right there. If you just Google DNA Barcoding 101, it'll send you right there. In addition, we've published this paper, Hyman et al. 2019, in course source. The DOI is linked there, but if you just Google cure-all DNA barcoding course source, this paper will likely come up. And there you can find detailed lesson plans, protocols, equipment lists, costs, slide decks, syllabi, basically everything that we use in this course in support materials if you want to try to implement this course or any sort of portion of it at your home institution. And please feel free to contact me if you have any questions or about how to do that. Lastly, I just want to do a plug for Cold Spring Harbor and the DNA Learning Center, that they have a lot of opportunities and educator training workshops. So definitely check out their website and see what sort of workshops are available to register for. And with that, I'll just say thank you.
Awesome. Thank you so much, Dr. Hyman. That was very lovely. So now we're going to transition to the breakout sessions for Q&A and further discussions. Breakout rooms are self-assigned and organized by speakers. So we will have one for Dr. Goodwin and one for Dr. Hyman, with Dr. Ray Enke serving as his co-facilitator. So I will go ahead and open the breakout rooms now for you all to select. So you should see a pop-up on your screen allowing you to select your own room. If you do not see that pop-up, you can navigate to the bottom of your screen and select the breakout rooms button and click the room you'd like to enter. If you're running an older version of Zoom and don't see the breakout room button, please post in the chat which speaker's room you'd like to enter, and some AAAS staff will assign you to that room. So this discussion is going to go on for about 25, 30 minutes, and we'll call everyone back together around 3:25. We'll give a 2-minute warning, and from there, we'll just do some final remarks, and that'll be it. So I hope everyone has a productive discussion, and we'll see you back here in a little bit. Thanks so much. All right, so everyone should be coming back from the breakout rooms now. Take another few seconds to everyone get back to the main room, but I hope everyone had a fantastic, wonderful discussion. Hope people's questions were answered, were able to connect, have some dialogue. So I'm wondering if Emma, Ollie, or Ray, if y'all want to take a quick minute to recap what y'all have discussed, or any final remarks you have before we get into some concluding thoughts.
Sure.
Yeah, sure.
Oh, go ahead, Ollie.
So we had some good discussions about implementing DNA barcoding. It was talking about some of the logistics involved, not only with the course itself, but also how you sort of pull it off administratively speaking. So those were some good conversations to talk through. We brainstormed some ideas for how you kind of do a DNA barcoding course locally, and do you really need to include a research question, and do you even necessarily need to include it in the sort of intro curriculum? Could it be more of something where it's like a one-off course before you get into the core biology course, and just a chance to sort of introduce students to biology and research in general before you actually get into the meat of the content that's really required to be a biologist in anyone's particular department. The last part was just sort of offering resources related to meta barcoding, which is really Ray's area of expertise, which is sort of leveling up from the intro individual species barcoding to sort of next gen sequencing, and some of the cool research questions that meta barcoding can address. Yeah.
Great. Thank you for sharing. Ray, anything you'd like to add? I feel like that covered it.
Yeah, I mean, that covered most of it. One other aspect we discussed was sort of the emergence of Oxford nanopore sequencing-based cures, and how that's starting to sort of revolutionize DNA sequencing-based cures. And there's some great educational resources out there. Oxford nanopore is just starting to get into the educational space, and they have a working group that is meeting, I think, twice a month. That you can find information through the Cubes Hub, and that's just another aspect of barcoding and meta barcoding to at least be aware of.
Thank you. Appreciate it. And Emma, how about your group?
We discussed some ideas on this Padlet. I just dropped the link in the chat. Some ideas that came up, I think there's a lot of interest in people being connected to resources specific to their discipline and their like community college versus 4-year intro bio versus advanced. So I'm not able to provide all of that on the spot, but there's definitely a lot of resources out there. We talked a bit about pros and cons of working in small groups versus the whole class on a single project. We talked about building iteration intentionally into the space of your course, having backup data for if students' experiments don't necessarily go out, but they still need to move on to an analysis phase. How students can disseminate information in different ways, micro publications being an awesome venue for publishing at a small scale. And it's one other thing I was going to hit, but can't think of it right now, so I'll stop the summary there.
Still a beautiful summary, so thank you so much for sharing. Great, thank you all for those recaps. and for the also fantastic presentations today. So before we go, I'd just like to thank our speakers and our facilitators, the rest of the AAAS IUSE team, and all of you who were able to join and participate in the workshop today, which is the last of our fall series and the final AAAS IUSE virtual workshop of 2023. We have a survey link, which we are posting in the chat and sending via email. That survey should only take a few minutes, and we really appreciate the feedback y'all have. And additionally, a recording of the presentation will be posted on our website and emailed to you in the coming days. Once again, thank you all for joining us today. Just a quick heads up with some few final announcements. We will be launching a call for workshop proposals later this fall, so be sure to keep an eye out for that. We hope some of you will consider presenting your work through our workshop series in 2024, and that all of you will join us for the series in the new year. And finally, for those of you interested in submitting a proposal for an IUSE project, or if you're an awardee and would like to connect with your NSF program officers, NSF has scheduled a series of office hours, which I've included on the slide here, for this fall and winter as a way for prospective applicants to communicate with the NSF program officers and to ask any questions they have about the program. You may find an introductory video explaining the office hours and the IUSE program, in addition to the office hours scheduled on the slide here, and also at that URL, and that's also provided on the NSF website. So if you were to Google NSF IUSE office hours, all that information should be there. And this slide is available in the deck, which we put it on the IUSE website, and the office hours and all this information we've also included on the AAAS IUSE site as well. So until next time, and as always, we look forward to engaging with you and learning from you, our AAAS IUSE community. Take care and enjoy the rest of the year. Thank you so much.